It's a really very complicated system, and we're only still just understanding it. When people say, well, why can't you just make a thymus in a dish? And I'm like, okay, I would do that if I knew what was in a thymus to begin with,
Hmm.
but we
Hmm.
keep finding new stuff.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I'm Kim Klonowski, your host for today's episode. The small organ that powers immunity in youth may hold surprising clues to staying healthier longer.
Our guest today is Dr. Nancy Manley, Director of the School of Life Sciences and the John O. Whiteman Dean's Distinguished Professor at Arizona State University. Prior to that, she was here at University of Georgia, where she served as the head of the Department of Genetics for a number of years. Nancy, welcome to the podcast.
Thank you very much. I'm very pleased to be here.
So, Nancy, the first question that we really like is asking folks about their pathway to their scientific career. So tell us how you ended up where you are.
Oh, what a great question. So I really started wanting to be a scientist when I was probably when I was in high school. I had the fortune of being in a public school system that had lots of science in my high school. And so I took all the biology courses and things that I could take, and I loved it. And I thought, I'm going to be a scientist. I didn't even think I wanted to be a doctor first, which I think a lot of people first start out wanting to be a doctor. I didn't want to do that. I had an opportunity in high school to spend some time working in a laboratory at a state lab that did genotyping for chromosomal abnormalities, which was really an unusual opportunity. And that just got me hooked on the whole idea of doing things in the lab. So I really started early on. When I went to college, I was a science major, but I was a biology philosophy double major for a long time. So I was always very interested in philosophy and ethics and things like that. And then when I had to make the final decision about what to apply for, I decided to just go get my PhD. And that was what I wanted to do, was to be a scientist. So it was a pretty straight path for me, not particularly convoluted. I started on it pretty early. So
Well, what are some of the major questions that you're asking in your research, Nancy?
So, my lab has always been very interested in understanding where the immune system comes from. What I like to say is that I'm a developmental biologist by preference. I was trained as a developmental biologist, but as soon as I started my own lab, I got interested in questions that were related to immunology. And so it took me years to even be willing to call myself an immunologist. And I'm still not really comfortable doing that, even though I've been doing it for 25 years. Developmental biologists ask questions about where things come from, what the origins of things are, how do they develop, how does what they do later on depend on what happened to them earlier in life, what controls all of that. So those are the kinds of questions that I like to ask. And the system that we ask them about is the immune system, specifically the thymus, which is the organ that everyone has, but most people have never heard of, and that organ is where all of your T cells come from, basically. And if you don't have T cells, your immune system doesn't work. And so understanding how we develop the organ that makes the cells that make your immune system work, I think is an interesting and fascinating question. And so the basic questions that we have always asked are, where does the organ come from? How does it develop? How does it function? And why does it stop doing that as you get older? The thymus has a really interesting life cycle of its own. And so understanding what it's doing at different ages postnatally in adults and young children, it does different things at different times in your life. It's not like another organ, right? So your heart always beats. It always beats, and its job is always to make sure that the blood is flowing through your body. And that's what your heart does for your whole life. And to a first approximation, your thymus makes T cells your whole life, but it makes different ones at different times. It's different sizes, it's got different dynamics, it responds to different environmental stimuli. It's most active when you're very young and then becomes less active when you're older. It's just has its own path. And so that made it fascinating for me as well.
So, to help folks picture this, how big is the thymus? Where is it?
Yeah.
So we know it kicks out T cells, but
Yeah, so that's really actually a really interesting question. So Your thymus sits on top of your heart. And when you're born, when human babies are born, their thymus is almost the same size as their heart. It's huge relative to their body size. When you're born, you don't have very many T cells circulating around in your body, in your lymph nodes, and your spleen and stuff. And that's because the thymus doesn't start kicking them out until in humans it's just before birth. And then most of them are made after you're born. So when you're born, you're relatively vulnerable to infectious diseases and things. And that's largely because your thymus hasn't made your T cells yet. So you're still making those T cells. As your body grows, your other organs grow with your body, right? So it's almost hard to think about it that way. But when your body grows, all of the parts of your body basically grow together. All your organs are the same relative sizes for most of your life, but not your thymus. Your thymus is biggest relative to body size when you're born. And then it continues to grow as your body grows, but not nearly as fast. And then it starts getting smaller and becoming less functional at a really early age when you're a child. So it's really only maximally functional when you're very small. And there's no other organ that's like that.
T cells aren't produced anywhere else. It's just the thymus. Oh, it's one of those answers.
It's one of those answers, right? So the answer is yes. Your thymus makes all of your T cells, except for when it doesn't. And there, and there, you can find weird T cells that are not completely like normal T cells. Say if you're looking in an animal model that doesn't have any thymus, there'll still be some things that look sort of like T cells, but they aren't really T cells. So I can say there are pretend T cells, maybe, if you don't have a functional thymus, but all of your functional T cells come from your thymus.
So I think we'll get into what those different types of T cells that are made at different times are. Before we do that, and maybe this will kind of solidify some of the points that David was alluding to, is what happens if you don't have a thymus?
So there are people who are born without a thymus. It's actually a pretty rare condition. But if you are born with no thymus, then you don't have a functional immune system. And you will be vulnerable to any infection that's around you. And it doesn't even have to be an infection that would kill you normally, that would be lethal normally, even things that could be relatively benign in people who have a functional immune system. If you don't have an immune system to protect you, then you're going to die. So, up until relatively recently, children who were born without immune systems, without a thymus, they just didn't live very long because it was impossible to keep them alive. So, it's important. You really must have it. Now, if you were able to be alive and be in an environment where you're never exposed to any kind of infectious disease. And we have mice in our labs that don't have a functional thymus, and they can live a normal lifespan, but we protect those mice so that they are in a very controlled environment so they don't get exposed to anything that could make them sick, because if they got sick, they would probably die. So you need your immune system to stay alive, and you need your thymus to have a functional immune system.
So, Nancy, how far back in evolution could we go and find a thymus? So, we know from what you just said that mice have them, but is it a mammalian thing or?
So it's a vertebrate thing. So birds, reptiles, mammals all have them. They're a little bit different in different animals, and we don't know a ton about that, but they all have a thymus and they have T cells and right. So the earliest that a thymus-like structure has been found evolutionarily would be lampreys. So lampreys are, pretty primitive animals, and they don't have a thymus, but they have cells that do some things that the thymus does. So that's probably the most rudimentary thymus in a living animal, but it's a vertebrate specific organ. Animals that are not vertebrates don't have a thymus or have a T cell based immune system.
So, Nancy, one of the main themes of your research, of course, is the fact that the thymus is making, it's not a static tissue. It's making different T cells at different times. It's not doing the same thing. We've talked about this in terms of the heart, as you gave it as a great example. and that the thymus is maybe making different T cells at different times. So, can you tell us more about that and why it may have some therapeutic implications?
Yeah. So, as I said earlier, when you're first born, you don't really have very many T cells at all because you're just starting to make them and export them out into your body. And so, newborn humans and other animals are very uniquely vulnerable to infection because of that. And so, they also don't have any memory. So, I'll take a step back, and I don't know if you've talked about memory on your podcast or
We
not.
haven't actually, so this is a good opportunity.
Yeah, so when you first make a T cell and it comes out of the thymus and it's there ready to do its work, it's called a naive T cell. And that cell is called naive because it's never seen the thing that it's ready to protect you against. It's ready, but it just has never seen it. And so it's naive, it's new. Once you are infected with something, then a subset of your T cells respond to that and they expand and they protect you and they fight the illness. And then when it's all resolved, most of those cells will die and go away. But a few of them you'll keep, and those will be called memory cells. So they're a special kind of cell that specifically remembers that it's seen that before. So, for example, for flu, the first time you get the flu, if you've never had the flu before, you'll get really, really sick, but your immune system will respond. The next time you get the flu's probably not a great example, but the next time you get the flu, your cells will remember, oh, wait, I've seen this before. I know what to do. And so it'll respond faster. And you may not even get sick the second time. So a lot of your immunity that you carry for diseases that you've had before, you carry because of those memory cells. But of course, to have a memory cell, you had to have been sick before. So babies have never been sick before. Everything's new for them, right? And so they need to have cells that are going to respond fast because there's no memory, there's nothing
Mhm.
else. And so they make a special kind of T cell that's a fast responding T cell. And those T cells can respond super fast if they get exposed to an illness to try and protect the baby quickly, right? In the absence of memory cells that are going to come in and do a really good job. So you need different kinds of T cells when you're born and you're totally never been sick before, right? Now, when you get older and you start to build up these memory cells, you'll keep memory for everything you've ever been infected to a first approximation. So then you need to you're building this functional immune system that's a mature functional immune system that can respond to old things quickly, but still has the capacity to respond to new things. You need a whole complement. Your immune system as an adult is not just one kind of T cell, it's lots of different kinds of T cells that all work together to protect you against infection, to protect you against autoimmunity, to protect you against all of those different T cells you need to make at some point, and then you keep them for basically your whole life until you're older, right? So your thymus has to make a whole bunch of different kinds of cells in order to make the full complement of T cells you need to have a fully functional immune system. The problem is that the environment in the thymus that makes a specific kind of T cell is different for different kinds of T cells. So it's really hard to make a whole bunch of different kinds of T cells all at once in the same organ because then you're having to make all these different kinds of cells and they all have different requirements. So instead, for the most part, what the thymus does is it makes one kind at a time. That's sort of an oversimplification, but like you make those really fast responding cells when you're a newborn. That's what you need in your periphery, and you
Mhm.
make those first. Then you make another kind of T cell that you need. So the thymus has to change. It's like retooling a factory, right? So you retool the factory a little bit, you make a new kind of cell. And then you make enough of those, you're like, okay, great. Now we're going to make this next kind of cell. So you retool the factory again, and it makes a new kind of cell. All the while, these cells are all going out, and then they live forever in your body. And so it's like it builds up this layered immune system so that you're making all these different times, but at different times, the thymus is tuned to make a specific kind of cell. So that factory analogy is really apt because you do have to retool the factory. You have to actually make changes to the organ itself in order for it to be able to make a different kind of T cell. So understanding how that progresses over time and what controls that is really important. So for example, if you take an older person, everybody's thymus stops working when you're relatively young for the most part. It'll still make a few cells, but it doesn't make very many, right? So you stop making those new naive T cells. And so once you stop making those, then it gets harder and harder for you to respond to new things that you've already seen because all your T cells are memory cells. They all remember old things, but they can't do anything new. So you might want to make a new immune system for somebody when they're older. Or somebody might go through chemotherapy because they have cancer. And the chemotherapy wipes out your cancer, but it also kills off a lot of your T cells. And you need to make some more, right? All kinds of things can happen. Or you may need a bone marrow transplant for some reason. And then part of doing the bone marrow transplant is you wipe out all your T cells first.
Mm-hmm.
So there's lots of therapeutic cases where you might do something to remove your current immune system, but then you need to replace it. Well, your thymus in an adult is not going to be able to do that by itself, not very well. It's not going to work very well. And even if you goose it up and make it work a little bit better, you're just going to make the T cells that adult immune system makes, that adult thymus makes. So all of those other kinds of T cells that you made when you were a kid in different versions of your thymus, you're not making those. So those are missing. What happens if those are missing? Well, one of the kinds of cells that you make only in a baby's thymus is a special kind of T cell called a regulatory T cell. That is a T cell that is specifically for protecting you against autoimmunity, for organ-specific autoimmunity. So, for example, lupus is a disease where organ-specific autoimmunity fails. But those special kinds of T cells, those special regulatory T cells, are only made in the baby thymus. So if you kill them all as an adult because you go through chemotherapy or something, and then you try to replace your immune system, you're not going to get those back because those only happen in a baby thymus. And then you're more vulnerable to autoimmunity. So that's just one example, but there's all kinds of different examples. If you're not working you're basically not playing with a full deck, right? You've killed off all of them, but you're only getting half of them back. And then your immune system's just not going to work properly anymore.
Talking about autoimmunity, we know that it's more common in women. And is that an issue with the differences in the thymus in males and females, or is it something else?
Great question. Actually, it's probably both the thymus and something else. I said earlier that your thymus starts to decline in function relatively early in life, and then it just declines until it's basically making very, very few T cells. And that happens during your juvenile years. And by the time, you are in your 30s, your thymus function is dramatically reduced. And by the time, you're as old as I am, you haven't had a functional thymus in a really long time. So you're basically stuck with whatever T cells you made earlier, which is fine because they live a long time and they're happy to be functional for decades. So that's all good. But that process of the thymus going from that really fully functional young child thymus to the really terrible, crappy old thymus that doesn't work anymore, that process is called thymic involution. And the rate of thymic involution is faster in males than it is in females. And that's because it's hormone responsive. So testosterone impacts that. And so what you end up with is young ages, male and female thymuses work about the same way. There's not any real different sex differences really early. But during that involution period, the male thymus crashes much faster, which you would think maybe that's a bad thing. Except if you think about it, if you're taking years longer to go from fully functional thymus to not very functional thymus, in between, the female thymus is making more cells. It's bigger, it's a little bit more functional, but that involuted thymus doesn't do a very good job of making T cells. And you're much more likely to make autoreactive T cells during that period. So women have this propensity in part because they tend to have more autoreactive T cells than men do, just because it takes longer for their thymus to involute. Now, there's a whole bunch of other things that impact frequency of and severity of autoimmune diseases, and different autoimmune diseases are different. But yeah, in general, women are more frequent. In some specific cases, men are less frequent, but have more severe disease. So it's a little bit more complicated than that. But yeah, the thymus is definitely part of it. And then part of it is other kinds of environmental impacts. We don't really know that much about how autoimmune disease starts and how to fix it, which is pretty obvious since such a huge percentage of humans have autoimmune disease and are not very well controlled. So this is still an area where we need an awful lot more research.
Yeah, I mean, I feel like we're talking about how important the thymus is in terms of protecting us long term, in terms of autoimmunity and all of these things. So it seems like a really important area therapeutically to get involved in. So why haven't we made more progress?
I think there's a couple of reasons why. First of all, it's a very finicky organ to work on scientifically, right? And it's partly because of what I said is like the thymus when you're young and the thymus when you're older and the thymus when you're a teenager, they're all functionally different from each other. And so it's hard to know, okay, let's make a thymus. Okay, which one do you want to make? You want to make a newborn thymus? You want to make a juvenile thymus? Do you want to make an adult thymus? What are you trying to accomplish? And if you want to make a full immune system, you really need all of those. So how do you do that, right? So I think that's actually a problem we've only recently known is even a problem because this knowledge about the different functional phases of the thymus, that's all pretty new stuff. That's not even
And
something that we've known for a long
And I
time.
do want to interrupt you before you finish your sentence, just to make sure that we're clear, is that what you make when you're young does persist when you're
Yeah.
older. So it's
You
not like
keep
it's
it.
just important when you're young. It
Yeah.
has a role later.
Right. So you keep all of those cells for your whole life. It's like you build it up over time, like building up a library, right? And so you keep all of those cells for your whole life, even the ones you made when you were a baby. So it's a long process, right? So therapeutically, you think, oh, great, we can just make a thymus organ just like we can make, they can make organs in culture now, right? And so let's just make one and then give it to people, and then they'll be able to make new T cells. And that would be a great idea. And there are actually a lot of people trying to do that. It's not like nobody's thought of that, but it's really hard to
Yeah.
do. And again, you're just making one kind. And so whatever you make from that is gonna not be everything you need, right? So there's other approaches too. You could make cells in a dish, and there's been some progress made therapeutically with generating systems in cell culture to make different T cells to mature. We can't do all of that in a dish yet, but people are working really hard on that. And that might end up being easier than trying to make whole organs, right? So if you could make cells in a dish that allowed you to mimic selection and things that happen in a baby thymus and a juvenile thymus and an adult thymus, then you could just grow a bunch of those cells, make a bunch of T cells of the different kinds you need, push them all together, and then inject that into a person. And because T cells live a long time, that might actually be a better therapeutic approach because then you can make sure that what you're giving them is the full complement of things.
This would be very individualized medicine, like specific. You'd have to get cells from the patient and make, I guess you're talking about organoids in culture,
You
or
actually can do a lot of it without organoids. You can actually do it just in monolayer cell culture too. But
okay.
a lot of people are trying to do organoids as well. But yeah, so you take your own bone marrow, so your own stem cells, and then put them into this culture dish, and then take them and put them back into you, and that would be fine. So I actually think if we were going to develop a therapeutic approach to try and help people, give people better immune systems as they age, that would be a smarter way to do it because it's a little bit easier to control. So that means that you're going to be able to know exactly what you're putting back in. If you put an organ in somebody, like I say, you can make an artificial heart. So they're trying to do that. That'll probably be a relatively early one that gets made that works for transplant. You give somebody an artificial heart. Well, you've made the heart and the heart can pump blood and that's all good. It's largely mechanical in its function. The problem with the thymus, you make a thymus and you think, okay, this looks pretty good. You put it in there. The thymus then has to generate all these new T cells. If it doesn't do that properly, you could give somebody autoimmunity because their thymus that you transplanted in isn't working quite right. And it's hard to know that from it's a much more finicky organ system to work with in that terms. So that's why I think the cell culture based approach is probably a smarter approach, but
Okay.
see what happens. People are working on both.
Yeah, and I think we're really focused on the T cell part because that's kind of the output, right? That's
Yeah,
that's
that's
the
what
immune system part. But, going kind of back to your developmental biology origin, this is all controlled by other cells. This is a lot of other cells that are kind of creating this environment, and they're all in different transitional states and different developmental states.
Yeah. So when I was talking about the factory building, which is the thymus, right? It's got a lot of different parts to it. There are multiple different types of cells in there. And they are doing very complicated things. And they're changing, their individual cells are changing their individual functions over time. The composition of the cells, the types of cells that are in there changes over time. Some cells come in from the outside and then go back out again. Sometimes they come in and they stay there for a while. There are these cells that are called recirculated cells, which are T cells that you make in the thymus. They go out into the periphery, and then for some reason they come back. And we don't know why they come back or what they're doing there, but they're there, right? So it's just all different kinds of cells. And it was only within the last, say, I don't know, 10 years when single cell technology became available that allow you to take complex tissues and look at the individual component parts of it based on a really comprehensive way of evaluating what their expression patterns and other things are. So you can identify every single individual cell and see how similar it is to other cells, right? So we may have thought before the single cell technology came out that there were like 5 different kinds of thymic epithelial cells, which is one of the kinds that we do. Now that we can do the single cell technology, we know that there are probably more like 15 different types of thymic epithelial cells. They don't necessarily look different, but they function differently. And that complement changes over time again. So you'd have different ones at birth than you do as adults. And some of that is because those cells change, and some of it is because some of the cells die and get replaced with new cells that do something different. It's just a madhouse in there. So it's it's a really very complicated system. And we're only still just understanding it. When people say, well, why can't you just make a thymus in a dish? And I'm like, okay, I would do that if I knew what was in a thymus to begin with, but we keep finding new stuff. So if we're still finding new things that are in there, I could make a thymus now based on what I know is in a thymus, but I guarantee you it would not be the same as a real thymus because I know there's stuff in there that I don't know what it is or where it comes from or how to make it. So it's a complicated factory, and the retooling the factories are real, like, because sometimes you need to change the machinery, sometimes you need to tune it up a little bit, sometimes you need to replace it entirely with new machines. It's like it's really functioning in a really complicated way.
So, Nancy, it occurs to me that as your research spans developmental biology and immunology, you're probably going to both developmental biology conferences, talking to one audience, and immunology conferences and talking to another. So, how do you bridge the gap? Do you have 2 completely different presentations, or how do you handle that?
Yeah, you do have to know who your audience is for sure, but I have a little bit of an advantage because neither immunologists nor developmental biologists know anything about the thymus for the most part. So, so usually when I talk to people, I just assume they don't know anything, and I can talk. The main thing is what vocabulary do I use, right? So the types of assays that we do that are common in immunology, I don't have to explain those to the immunologist, but I might have to explain some of the developmental biology terminology and concepts to them. But yeah, for the most part, I work on something that very few people work on. And so I have to always explain it to everybody from scratch.
So, Nancy, you were a previous era colleague here at UGA and you moved to ASU and you took on an administrative role. So, I thought maybe we could take a little bit of time talking about that transition as well and how that's been for you.
Oh, yeah. So when our daughter went away to college, we decided that my husband wanted to retire. And I was like, okay, let's look for a new opportunity. I did a lot of stuff when I was at UGA, but I felt like, I could do more stuff if I went somewhere else. And so I started looking for a job and I figured that I could just keep running a research lab and getting federal grants and doing experiments and writing papers and things. But at some point I was going to have to stop doing that because at some point I was going to retire, right? or die. One of those 2 things was going to happen to me, right? And so I figured I would rather shut down my lab in a normal sort of controlled fashion and move into working in an administrative role where I could still be involved in science and in helping graduate students and in running educational programs and things like that, but we'd be doing it on a larger scale and not really running a research lab. So my original plan was that I was just going to shut my research lab down entirely and not do any more research of my own. And that failed miserably because I still run a research lab. So but it's much smaller, right? So I used to have a very large lab with lots of people and lots of projects and things like that. And now I have a much smaller lab and I work on much more focused questions. And for the most part, my lab is composed of me and a senior research scientist who has worked with me for like 15 years and my mouse technician who's worked with me for 12 years and that's it, right? So it's much smaller scope than what I was doing before. Although I did just agree to take on a graduate student, which I also said I was not going to do again. So I was talking to one of my colleagues today and I said, okay, I just need space for my research scientist. Okay, now I need to have, I'm taking this. He was like, so you just lie all the time, right? You said you weren't going to do any of these things. I was like, well, I thought I wasn't, but it turns out that it's very hard to quit being a scientist. And I like it. So it's not that hard to convince me to do it.
Right. But I imagine there were also some reasons why you wanted to take on the role you wanted to take on, right? I feel like you probably had some ideas about how to restructure how science is done or just reinvigorate, just lots of ideas.
Yeah, I find that working in academic administration, which is, being in an administrative role in an academic institution, has a lot of rewards for me. It's not for everybody. I guarantee you it is not something everybody should do. But that's true for most jobs, right?
Hmm.
Most jobs you shouldn't do it if you don't want to. The thing that I really like about academic administration is that I can make a difference and promote the careers of many, many more people, right? So at ASU in particular, because it's such a large university and the School of Life Sciences is a very large school within ASU, I have the ability to support and promote the future careers of thousands of people. And if I'm just running my own research lab, then I'm supporting the careers of the few people who work in my lab. And sometimes that's, 12 or 15 people, which is a big lab for me, but it's still not thousands of people, right? And I really get a lot of satisfaction from being able to support and promote the careers of those people, not just by helping them individually. For the most part, I don't help people individually. What I do is that I create systems and support programs and develop educational programs and degree programs that facilitate large numbers of people being able to realize their goals and find what they want to do and find their passion in science. And that's really satisfying. I find that to be really, really satisfying. And so when I took this job on thinking, okay, for the last years, however long that is of my career, this is what I'm going to mostly do. It's still what I mostly do, as I mostly am still administrative. The science is a fun thing for me to do, but I'm still probably 80-90% administration. The rest of me is spending time running this large school, making sure that we have thousands of undergraduate majors and hundreds of graduate students and 9 different PhD programs. And it's just a much bigger scale. And it really lets you think about things in new ways and implement things in new ways and support interdisciplinary research and do things like that you can't do just in your own single lab in your own research career. And so I've just found that to be very, very exciting.
So, Nancy, last question. When you're not thinking about the thymus and not carrying out your administrative duties, what are you doing in your free time?
Yeah. So I hesitate to say, my biggest sort of hobby that I have is actually an online gaming club that I'm part of.
Oh, cool.
Nice.
So I'm actually the president of an online gaming club for SimCity Build It, which we can leave the name of the game out when we do it. But yeah, I have an online gaming club that it's like
Oh,
this
that's cool.
international. I've got people all over the world who are in the club and we all interact online. It's really fun. So that's what I do when I'm not doing science.
So you also administer an online gaming
I
community.
do. I just can't seem to
She
not
can't get away
do
from it.
Yeah, we had another club president and she left and then it was like, oh, somebody else needs to do it. And everybody was like, you do it. And I was like, really? Okay.
Well, our guest today is Dr. Nancy Manley, Director of the School of Life Sciences and the John O. Whiteman Dean's Distinguished Professor at Arizona State University. Nancy, thanks for being with us.
Thank you so much for having me. It was a lot of fun.
Yeah, it was. Links to additional information about Dr. Manley's research can be found on our Instagram account at PPP Podcast UGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out our previous episodes. Please rate the podcast. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It's supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemeth, for research, editing, scheduling, and keeping us on track.